All right, welcome back. So last time we left off talking about tonicity and diffusion. So those, or not those, diffusion is a type of passive transport. And so remember we talked about there are two types of transport, two types of ways to get things from one side of a cell membrane to the other. So for example, to bring something from outside of the cell to inside of the cell. We can do that passively, which means that the movement of whatever that object is or molecule is, so water, for example, from outside the cell to inside the cell doesn't require energy. As opposed to active transport, the subject of this slide. So active transport means that moving that molecule from outside of the cell to inside, or vice versa, requires energy. And so remember, one of the things we talked about last time was the energy molecule, which is adenosine triphosphate, or ATP. And that effectively what happens is we use enzymes, ATPases, to break down the chemical bonds in ATP, specifically the bond between the last two phosphates. And when we break that bond, that gives off free energy, and that allows us to do cellular work. So we're going to talk about ATP quite a bit throughout the course, but I figured I should probably mention when I talk about requiring energy, what I mean is that we're going to have to break down ATP in order to perform this work. So active transporters can be thought of as being solute pumps. Throwing it back to last time when we talked about two terms, solute and solvent. And remember that the solvent is a fluid, and the solute is the thing dissolved in that fluid. And so in terms of active transporters being solute pumps, we're going to pump things dissolved in fluids, so things like sodium, potassium, we're going to talk about that quite a bit, calcium, glucose, we're going to pump those from one side of the cell's membrane to the other, and we're going to use these transporters to do it. So the reason that this active transport requires energy is that we are pushing molecules against their concentration gradient. So for example, the concentration of sodium outside of a cell is typically much, much higher than it is inside of a cell. And so one of the things that cells have to do is to minimize the amount of sodium inside of them, because if we just allowed lots and lots of sodium to come into the cell, well then we run into some of that, some of those tonicity problems that we talked about last time, where that'll cause cell swelling and potentially the rupture of cells. So in order to ensure that the cells don't swell too much, they spend a lot of energy kicking sodium out, or to stick with the terminology that I'm using here, they spend a lot of energy, a lot of ATP, pumping sodium from inside of the cell to outside of the cell. So they're pumping it from inside, where there's relatively little sodium, to outside, where there's already a lot. And so again, they're pumping it against their concentration gradient. They're actually pumping it from low to high concentration, which is against that concentration gradient. So to do that, effectively you're kind of pushing sodium uphill there. And so then that requires energy. So going against a molecule's concentration gradient requires energy, means it requires active transport. So there's two types of active transport that you can see there. The first is primary active transport. And so in the case of primary active transport, the energy to do that work comes directly from breaking down ATP. And so what you've got here, so this picture on the left, that is primary active transport. And so what we're going to do is we're going to break down ATP. We're going to break the bond between those last two phosphates. And then when we do that, giving off free energy is going to allow us to pump sodium outside of the cell. And then we also simultaneously pump sodium inside of the cell. So in primary active transport, we break down ATP to eject something from the cell. So that's what that is. Secondary active transport, essentially what we do is we take advantage of that gradient that we have created, or that gradient that we have exaggerated, and we use that gradient to help us do extra work. So now that we've created this really substantial gradient where there's lots of sodium out here, remember that the bigger the concentration difference, the more sodium out here there is relative to what's inside of the cell, the more the sodium wants inside of the cell, effectively, the more readily it'll diffuse back inside of the cell, given that ability. And so we can allow sodium to flow back inside of the cell, and then we can use that, it going with its concentration gradient, to drive something else either into or outside of the cell. And so those are what symport and antiport are. And so I'll explain that in a little bit more detail because the next 2 slides cover both of those things. So here's our primary active transport. So there's some terminology on there that I should explain. So hydrolysis means to use water to cut. And so when we break down ATP, when we break that last phosphate bond, we use water to do it. So hydrolysis of ATP, from a practical standpoint, just means that we're breaking down ATP, which gives off energy. In terms of phosphorylation, that means that we add a phosphate. And so when we break that phosphate, that last phosphate off of ATP, we can add it to this sodium-potassium pump here, which is also known as sodium-potassium ATPase because it's an enzyme. And so we can add that phosphate group, so that's phosphorylation, to this sodium-potassium ATPase, also known as a sodium-potassium pump. And when we do that, when we add that phosphate group and give off that energy, that causes that pump to change shape. And so when it changes shape, it's going to, again, expel sodium, but then it'll allow potassium to come back inside, which is why they're called sodium-potassium pumps, because they do both things simultaneously. They kick out the sodium, and then they allow the potassium to come back inside. By the way, potassium is also moving against its concentration gradient. So there's typically quite a bit of potassium inside of the cell, relatively little outside of the cell. So we're going to pump both these things against their gradient. We're pumping sodium to where there's more of it. We're pumping potassium to where there's more of it. And so you can see that there's 10 times higher sodium in a plus of sodium outside of the cell. But because of that gradient that we've created, given the opportunity, they're going to leak back in. So there are different channels that they can leak in through. But they're actually going to, sodium is going to leak in, and then potassium is going to leak out. So potassium there is K+. So what you need to know in terms of primary active transport, again, we're going to break down ATP, and that's going to allow us to pump something, often sodium, against its concentration gradient. And again, once we've established that gradient, or once we've expanded on that gradient, we can then use it to allow us to perform another type of work. So this is Hoover Dam, and so my analogy here is that the dam itself is the cell membrane, and so all the water back here in Lake Mead behind the dam, that's your sodium, and so we can allow sodium to move back across the membrane, or in this analogy, through the dam, and in doing that, that will perform some other type of work. So for example, sticking with Hoover Dam, when we allow the water to move through the dam, it's going to turn turbines that generate electricity. That's one of the ways, I think the primary way, that Las Vegas is powered, right? So we can use that gradient, or that energy, stored energy from water here, to produce electricity. In the case of a cell, we can use that energy of all this sodium out here to flow with its concentration gradient, and then that can do other types of work for us. So, for example, and if you hear my dog barking in the background, I'm sorry, the UPS guy just showed up. For example, in the intestinal cells, one of the things that we can do is, so you'll have, in terms of what you're looking at, it's the same basic scheme right here, right? Where you've got the extracellular fluid out here, which is the lumen of the intestine. It's the inside of the intestine. And then you have the cell membrane of an intestinal cell, and then the inside, the cytosol of the intestinal cell. So you got lots of sodium out here in the lumen of the intestine. In addition to that, some of the glucose that we ate. So let's say you had something with both salt and sugar in it. Sounds like a decent meal, right? And so what we'll do is we will take advantage of this gradient where sodium really wants inside, and we'll let it inside provided it brings glucose with it. And so that's taking advantage of that gradient that we've created. And so one of the things we'll actually do, we'll talk about this later when we get to the intestines, is we'll actually actively pump out sodium from the bottom side of the cell to keep the concentration of sodium inside of the cell really low so that we can continue to pump things through. We can continue to take advantage of symport. So in terms of symport, we're going to allow something inside of the cell, but it's going to bring something with it. So for example, sodium, we're going to allow it inside of the cell. We're going to allow it to diffuse with its concentration gradient, but it needs to bring glucose with it. And so that's symport. So the short version is, in symport, both molecules go the same direction. They both go from inside or outside of the cell to inside of the cell, or vice versa in some cases. Antiport, so if you're anti something, you're against it, right? So we'll talk a lot about antagonists. Somebody, in the case of literature, that's the bad person in the story, right? So antiport, you're against something, and so antiport is opposite direction transport. And so what happens there is we allow sodium to move with its concentration gradient, but then we expel something else. So the two molecules move in different directions. So that's one of the ways that cells can maintain their pH, maintain the acidity or alkalinity of the cell, is to regulate the amount of hydrogen inside of the cell. So we can allow sodium in and then simultaneously kick hydrogen out, and so that's antiport. Again, the two move in opposite directions. All right, so as long as we're talking about bringing things into a cell, we should probably talk about vesicular transport. So what that pertains to, so this is again kind of your generic cell, right? And we've taken out all the organelles, but here's the extracellular fluid, here's the outside of the cell, and here is a basement membrane or something underneath this cell. So one of the areas where we're going to talk about moving things across the cell pretty frequently is in the case of the intestinal cells. And we'll also talk about it in the context of cells that line the blood vessels, which are called endothelial cells. So in terms of bringing things inside of a cell, there are essentially three things that we can do with it, but they all, the molecules that we bring in move inside of vesicles. You can see vesicular transport here. So what is a vesicle? A vesicle is basically just an extension of the cell membrane. The membrane kind of bubbles in on itself and then wraps around whatever the item of interest is that we want to bring inside of a cell. So that could include things like different hormones. In the case of some of the cells of your immune system, that might include like a bacteria that those immune cells attack. So there's a number of different things that we can bring inside of a cell. So endocytosis then, as you can see there, is moving things into a cell. And then phagocytosis is cell eating. And so phagocytosis, that goes with that example that I just talked about in terms of cells of your immune system actually like wrapping around a bacteria, or they wrap around other things. So that includes things like if you're exposed to asbestos, or if you, really fine particles, plastics and stuff, they'll wrap around that to try to help you dispose of it. So I've got a little bit more detail on vesicular transport here on the next slide. So again, sticking with endocytosis, what that means is just bringing something inside of a cell. And there are subcategories of that is kind of a way to think about it. So one of those is penocytosis, which means cell drinking. And so you see this, for example, in the cells of the intestine, where they'll actually just kind of bubble in a little bit, and they'll collect some of that extracellular fluid as well as some of the small molecules that are in that, and then they'll transport that inside of those intestinal cells, where then they can break down any proteins that are in there. They can transport things across through the cell into the basement membrane, which, before I forget, that is transcytosis. So whoops, I'll show you my gross picture in a second. So transcytosis is just moving something from one side of the cell to the other. And so that's kind of what you've got pictured here. So we move something, so again, you're gonna see that in the cells that line blood vessel walls, again called endothelial cells. We'll bring something inside of the cell, we'll actually transport it across the cell and then kick it out the other side. That is transcytosis. In terms of receptor-mediated endocytosis, so cell drinking just kind of, the cell membrane bubbles in and just captures whatever is nearby. It's not very selective. It's just, whatever happens to be floating by, we're going to transport that inside of the cell. And that includes fluid, which is why it's referred to as cell drinking. That's as opposed to receptor-mediated endocytosis. There's something, essentially a lock, if you will, on the membrane of the cell. And so whenever the key matches up with that lock, then we'll transport whatever that molecule is that's attached to the key inside of the cell through endocytosis. And so you can see there that that's pretty selective. So for example, we use that with insulin. So insulin will attach to cells and then be taken inside. That falls under that category. We also see that with various pathogens. So pathogens are things that make us sick. So things like the flu virus, diphtheria, and cholera. So diphtheria is a bacterial infection that you probably haven't heard of except in the concept of vaccines or inoculations. So diphtheria is a respiratory bacterial infection. It was a real scourge, particularly in kids, in the early part of the 20th century. So, for example, there were some particularly bad outbreaks in the early 1920s. So, as an example, in 1921, there were almost 16,000 deaths as a result of diphtheria. And so it is spread how we initially thought COVID was spread, which is through droplets. So if you like cough or sneeze, and let's say you sneeze into your hand and then you go touch a door handle or something like that, and somebody touches it right after you, they're going to have those droplets on their hand, and then if they touch their face, then they can be exposed to that bacteria and get that infection. And so once those diphtheria bacteria are inside of the respiratory tract, then they can bind to, using, trying to highlight what I'm still talking about here, they can use receptor-mediated endocytosis. They can bind to the epithelial cells, which are the cells that line your respiratory tract, and then by looking like things that the cells actually want to bring inside, the cell will transport that bacteria inside of itself, where the bacteria then hijacks the reproductive machinery and the protein building machinery of the cell, makes a bunch of copies of itself, and then explodes, and then you've got a bunch of bacteria, right? And so inside of the respiratory tract, so think like inside of your nose, in your throat, when those bacteria do that and make lots and lots of copies of themselves and kill lots and lots of cells that line your respiratory tract, well, then you get this kind of accumulation of all of those dead cells, and that results in a sort of gray covering inside of the nose and the throat, and then potentially in the lungs if that bacterial infection gets far enough down in there. And so here's my gross picture. It's coming up, so just so you're ready for it. So you can see this kind of gray covering here in this person's throat. And I'll click that off. So that's a person with a diphtheria infection. And so then what ends up happening is that accumulation of dead cells and debris, etc., eventually kill through asphyxiation. You can't breathe. It basically clogs up your respiratory tract. And so until there was a vaccine developed for that, that was a pretty deadly condition. Other things that attack the digestive system, cholera, you've probably heard of. It's a waterborne bacterial infection that comes from water that is contaminated with fecal matter. And basically what it does, it attacks the lining of your intestines, and in some people can be asymptomatic or can produce really mild symptoms, but in a few people it can produce really severe symptoms, and basically it produces such severe diarrhea that people can die within a few hours of dehydration, of starting to become symptomatic. And so again, the cholera bacteria uses the same method to transport itself inside of cells. And then, to no one's great surprise, right, COVID-19 does a very similar thing. So the particular receptor that COVID-19 uses is an enzyme called ACE2, which is an enzyme that's also found throughout the respiratory tract, but it's found in not only throughout the respiratory tract, but also in the myocardium, so in the heart as well, which is why you've seen some of those stories about, particularly early in the pandemic, about athletes having heart issues, right, is because of the receptors that once the virus spreads throughout the body, it eventually can make its way to the heart and then cause some heart problems. So COVID spreads the same way through this receptor-mediated endocytosis. It hooks onto those ACE2 enzymes on the surface of the cell, tricks the cell into bringing it inside, and then produces lots and lots of itself. And then so that produces the systemic infection and inflammation. All right. Well, endocytosis, as mentioned, is coming into a cell. Exocytosis, as you, I'm sure, are aware, or at least would have guessed, is going the other way. So exocytosis is leaving a cell or ejecting material. So why do cells eject things? So the two most common signals that cause cells to release things are that a hormone binds to them. So for example, there are a type of hormone called tropic hormones. And so those are hormones that cause the release of other hormones. So for example, you're probably familiar with human growth hormone. So growth hormone in a sports context does things like increases muscle mass, decreases fat mass, those kinds of things that can be beneficial to performance. That's what drives those growth spurts in kids up until puberty. And so growth hormone, its release from the anterior pituitary, which is part of your brain, that release is initiated by a precursor hormone, which is called growth hormone releasing hormone. And so you've got when the anterior pituitary receives growth hormone releasing hormone, which is hormone binding, then it releases growth hormone. And so that can be one of the things that can initiate the ejection of something, in this case a hormone. But what we're going to talk about, the ejection of material most commonly is related to the charge of a cell. So, and we cover this a lot in Structure and Function 1, but when you change the charge of a cell, so at rest, cells are generally negatively charged. And so, and I say generally just to hedge my bets, all of them that I'm aware of are negatively charged. But at rest, they're generally negatively charged, and that includes both neurons, so nerve cells, and muscle cells. And so when we get a change in charge, we'll stick with neurons for now, that'll cause a neuron to release neurotransmitters. So things like acetylcholine, which then causes skeletal muscle contraction, or they'll release other hormones like GABA and glutamate that can cause stimulation or inhibition of of neurons that they communicate with. So, and we'll talk about mucus here in a second. So exocytosis is ejecting material, typically happens, or we're going to discuss it happening in response to hormone binding or a change in charge of the cell. And so what this picture is, not that I expect you to know it, the slide looked a little plain, is ejecting something, right? And so that's inside of, let's say, the end of a neuron. You're going to have these little vesicles that are going to contain acetylcholine. We'll go with a motor neuron. And so in response to that change in charge, they're going to migrate to the end of the neuron, so the axon terminal, and then they're going to bind there and they're going to release their acetylcholine, which will then be picked up by the muscle cell, cause it to change charge, and then that'll cause muscle contraction. All right, so the cytoplasm. So as you know, the cytoplasm is inside of the cell membrane. It's this semi-fluid jelly-like material there inside of the membrane. And so there are essentially three categories of things in there. There's the cytosol itself, or the cytoplasm. So cytosol, cytoplasm, I'm going to use synonymously. They mean the same thing. So you've got that jelly-like material, and then you also, in some cases, you have inclusions. And so what are inclusions? Inclusions are things that could be included or could not be included. So for example, one of the inclusions that we're going to talk about quite a bit is glucose or glycogen, right? And so we store glucose inside of liver cells, and we can then release that glucose if our blood sugar drops too much, so if you've gone too long between meals. Or in the case of muscle cells, we can use that stored glucose, that glycogen, to help fuel muscle contraction, to provide the energy to help us remake ATP. So glucose or glycogen would be an example of an inclusion. So again, something that may be in a cell or not, but it depends upon what particular type of cell we're talking about. So another type of inclusion is lipid droplets or fat droplets, which are inside of your adipocytes, your fat cells. That's also an example of an inclusion. In addition to that, you've got your organelles. So your organelles are your cellular machinery that do all the things that the cell needs to do in order to continue to live and to function. So that includes things like the nucleus, which contains the DNA, but that also includes things like mitochondria, the endoplasmic reticulum, Golgi apparatus, etc. Speaking of the mitochondria, so anytime I ever ask in an in-person class what people know about the mitochondria, people always say the powerhouse of the cell, which is great. Like, that's a good thing to know about it. That's a good summary of what the mitochondria is, because that's really what it does. But what does it mean that the mitochondria is the powerhouse of the cell? Well, the short answer is it means that that is where we use oxygen to remake ATP. So that's where we're going to make the vast majority of ATP. Again, the energy molecule that drives our sodium-potassium pumps. It's the energy molecule that drives muscle contraction and a bunch of other cellular processes. So we make that ATP, or remake that ATP, inside of the mitochondria. And so that's why it's known as the powerhouse of the cell, is because that's where the vast majority of our ATP production comes from. So we can, in certain cells and under certain conditions, produce some we can produce some ATP in the cytosol of the cell. So for example, your muscle cells, particularly your fast twitch muscle fibers, are able to produce some ATP in the cytosol without using oxygen, but the vast majority of our ATP supply comes from the inside of the mitochondria. In terms of the density reflecting a cell's energy requirements, so cells that have higher energy requirements have more mitochondria. So for example, your liver cells and your kidney cells, those are cells that are always active, right? Other than like during intense exercise, they become less active during intense exercise, but in general, they're always pretty active. And so because those cells are always going, they're going to have lots of mitochondria. Same thing in your myocardium, in your heart muscle. Heart muscle, because it's always going until you're not going anymore, has to produce lots of ATP. And so your cardiac muscle, your myocardium, has lots of mitochondria as well. And we can see that in skeletal muscle. So skeletal muscle is your voluntary muscle. And so there's two broad types or two broad categories of skeletal muscle cells. There's slow twitch fibers and fast twitch fibers. And you use your slow twitch fibers to do most of your activities of daily living. So, propelling yourself from point A to point B, lifting something off a shelf, writing, typing, all those kinds of things. That's your slow twitch fibers doing that. So those fibers tend to be recruited much more often than your fast twitch fibers. So we use fast twitch fibers for generally big explosive things. So when you're walking, you're using your type 1 fibers. When you're running, you're also using your type 2 fibers. So we don't recruit the type 2s as much, so they don't have as many mitochondria. Your type 1s, your slow twitch fibers, because we use them all the time, they're going to have lots of mitochondria. And in addition to that, because I think this is interesting, the number of mitochondria in a cell, particularly in skeletal muscle cells, is adaptable, right? And so if we have, let's say if you're sedentary, you don't really do anything, and then New Year's resolution, you've decided you're going to take up an exercise training program, and you exercise for several weeks. At the end of that program, or after the course of several weeks, the trained muscle cells, so let's say you went from being sedentary to starting to get ready for a 5K. So the muscle cells that you've used in your training are going to have changed. And one of the ways that they're going to have changed over the course of those several weeks is that they're going to add more mitochondria. And the mitochondria that they have are also larger. And by having more and by having larger mitochondria, they're going to be better able to produce more ATP and to do that more quickly. So you can see in terms of the mitochondria, so that there's two membranes. There's the outer membrane and there's an inner membrane. And because it relates to what we talked about earlier, so remember we talked about active transport and creating gradients. So one of the way that we produce most of our ATP is basically we pump hydrogen into this space between these two membranes, and then we allow hydrogen to flow back inside of this inner membrane with its concentration gradient. And when we do that, runs through an enzyme called ATP synthase, and then that allows us to produce ATP. So we actually create a gradient of hydrogen in this case, rather than sodium, but we create a hydrogen gradient out here that helps us, we take advantage of that gradient as it moves back inside of that inner membrane to produce more ATP. And we'll talk about that a lot more at the end of the class, but because it's an example of creating a gradient and taking advantage of that potential energy, I wanted to mention it. So the other thing I have on there is inside of this inner membrane is where we're going to break down parts of carbohydrate. So CHO, if you see that abbreviation on any of my slides, I'm referring to carbohydrate. Fat is also CHO, but kind of in general, if somebody says CHO or includes CHO in writing, they're referring to carbohydrates. So that's where we're going to break down part of our carbohydrates is inside there, as well as fat and protein. So when you eat something, to degrade it to ultimately reproduce ATP, most of that work in degrading that food late in the process comes from or happens in the mitochondria. All right, other things, other types of organelles. So we've got our ribosomes. So ribosomes, the short thing or the bullet point thing that I want you to know about ribosomes is that they synthesize proteins. And we're going to talk about this quite a bit more in the third part of this talk, which when we cover DNA and RNA, we'll talk about ribosomes quite a bit more. But again, the primary thing I want you to know about them is that they synthesize, they build proteins. And because they build proteins, ribosomes, like mitochondria, are also responsive to exercise stimuli. So if you have a trained muscle cell, whether it is endurance training or strength training, you're going to need to produce additional proteins in response to that exercise. So endurance training, as mentioned, you're going to need to produce more mitochondria, and so we need to build some proteins to do that. Strength training, especially if you're engaged in heavy strength training, you're going to produce more of the contractile proteins, more actin, more myosin, so we're going to need ribosomes to do that as well. So trained muscle cells then are going to have more ribosomes than untrained cells. They're going to be better at building proteins. In terms of where ribosomes are found, they can either be free ribosomes, which means they just kind of float around the cytosol or the cytoplasm, or they can be membrane-bound ribosomes, which means that they are embedded in the endoplasmic reticulum, which is our next thing here. So the ribosomes that are bound to the endoplasmic reticulum primarily synthesize proteins that are intended for, we'll stick with that. They're intended for export from the cell, things that we're going to kick out of the cell. So the endoplasmic reticulum is this blue stuff here. So the purple thing there is the edge of the nucleus. As you can see there, it's the nuclear envelope. And so that is continuous with the endoplasmic reticulum. So there's two types of endoplasmic reticulum, or ER for short. Rough ER, which means that there are ribosomes embedded in it. And then smooth ER, which means that there are not ribosomes embedded in it. So rough ER is going to create proteins, because again, that's what ribosomes do. So as you can see there, it's well developed. There's lots of it in cells where we secrete things. So you see rough endoplasmic reticulum, a well-developed or expansive rough endoplasmic reticulum in cells like goblet cells. So when we talk about the respiratory tract, goblet cells line your respiratory tract and they produce mucus. And so they are going to be producing mucus all the time, right? And so one of the things that mucus does is helps trap things that we breathe in. So whether that's dust or invading pathogens like bacteria or viruses, they produce the mucus that helps trap that. And so because they're doing that all the time, they have lots of rough endoplasmic reticulum to produce lots of the proteins that help make up mucus. Similarly, liver cells are going to produce bile, which helps us in the digestion of fats. And so they're going to be producing bile fairly consistently, those liver cells. And so again, they're going to have a really well-developed rough endoplasmic reticulum. The smooth endoplasmic reticulum, as you can see there, it's only found in a few types of cells or a few tissues. So where we find that, for example, there's a well-developed smooth endoplasmic reticulum in the testes because it helps us synthesize steroid hormones like testosterone. We also find lots of smooth endoplasmic reticulum, as you can see there, in the liver and kidneys because smooth ER helps to detoxify drugs, helps us break down pesticides, and helps us neutralize cancer-causing chemicals. In the case of the liver, it also plays a role in breaking down glycogen, which again is stored glucose. So liver glycogen is what we use to keep our blood sugar within normal limits. So other than those few tissues, though, we don't really have smooth endoplasmic reticulum in very many other places. There is some in intestinal cells because they play a role in the transport of fat, so transcytosis, moving it from one side of the intestinal cell to the other, but by and large, smooth endoplasmic reticulum is not found in that many different cell types. All right. The endoplasmic reticulum works together with the Golgi apparatus, this organelle here. So the proteins that are produced by the rough endoplasmic reticulum then are transported to the Golgi apparatus, which, as you can see there, takes those proteins, it modifies them. And so basically what it does is it can change those proteins up. It can trim off sugars, it can add sugar groups, it can trim off phosphates or add phosphates. A variety of different modifications can occur inside of the Golgi apparatus. And then what it's going to do is to package those proteins inside of a vesicle for transport either out of the cell or maybe across the cell. So we see that in the case of lysosomes, which are the cell's stomach that we're going to talk about here in a second. So the thing to know about the Golgi apparatus is that it takes proteins from the rough endoplasmic reticulum. It can modify them, concentrate them, and package them so that then we can either export them, which is what you see happening in pathway A. So you see that in the case of neurotransmitters, which again are chemicals that neurons or brain cells, nervous cells, use to communicate with each other. And so we'll export those, we'll release those through exocytosis. We can also generate just more parts of the cell membrane. So that's what you're seeing here, this little empty vesicle. They can generate those vesicles because, again, they're going to have that phospholipid bilayer to help repair and maintain the cell's membrane. And then the last thing there is that they can become part of a lysosome, which again is the cell's stomach, which is the last thing we'll talk about here in a second. All right, so peroxisomes. So peroxisomes are found in a wide variety of cells. We're going to talk about them primarily in their context in the liver. But what do peroxisomes do? So they use oxygen, that's O2, to detoxify things. So for example, in the liver, peroxisomes are going to play a role in helping us to break down alcohol. So if you consume alcohol, part of the metabolism process is going to occur in the peroxisomes inside of the liver cells. Kidney cells also have quite a few peroxisomes to help detoxify. So the kidneys play a filtration role, as we'll talk about. And then peroxisomes also help us break down free radicals. What are free radicals? The definition is that they are highly reactive chemicals with unpaired electrons that can attack cell membranes. So free radicals, what they are, think back to your high school chemistry class. Remember that if a molecule has unpaired electrons in its outer shell, it's an unhappy molecule. It wants to be happy. It wants to have an even number of electrons in the outer shell. And so what that free radical with unpaired electrons will do is it'll try to steal electrons from somewhere else. So basically it'll attack the cell membrane and then to steal those electrons, which can then damage the cell membrane. And if you have enough of that damage, enough free radical damage, that can result in the death of the cell. And so peroxisomes help make sure that doesn't happen. So where do free radicals come from? In a metabolic context, most of them actually come from the energy production process, or the ATP production process, to be more specific. So the end result of making ATP in the mitochondria is that we are going to attach oxygen to two hydrogens, and then we're going to make metabolic water, right? And so that's how we get rid of the hydrogens that we pull off of the carbohydrates, fat, and protein that we consume. But that process of making metabolic water, H2O, doesn't always go entirely correctly. So sometimes you get things H2O2 and various other different chemical iterations, and so some of those are free radicals. So basically, free radicals are most commonly produced in the mitochondria as basically mistakes of the ATP production process. And so when those mistakes happen, the peroxisome's job is to go through and neutralize those free radicals to make sure they don't do a bunch of damage to the cell from those mistakes that were made inside of the mitochondria. And then our last organelle is the lysosomes. And so the lysosome is on the picture there. As mentioned a couple of times already, those are essentially the cell's stomach. So they're little sacs of digestive enzymes. What they do is to pull hydrogen inside of themselves, and so they are very acidic. They pull hydrogen from the cytosol, and so the reason I keep referring to it as the cell stomach is because they are acidic on the inside, just like your stomach is. So they play a variety of important roles. So in immune cells, where we talked about phagocytosis, where immune cells will take in bacteria or other invading pathogens. What we then do with that bacteria is we actually push it into or move it into the lysosomes of those phagocytic cells, and then that really acidic environment will kill them, right? And so lysosomes then can help us in immune cells to neutralize invading pathogens. We also use them, we'll talk about them a lot in 201, so in instruction function 1, in the context of you've got a type of cell inside of your bone called osteoclasts. And osteoclasts, what they do is to break down bone. And so they use the enzymes inside of the lysosomes, that really acidic environment, to break down bone to release calcium, to help keep our blood calcium up, but also as part of the normal bone remodeling process as well. So lysosomes also play a role in cell death. So for example, in the absence of oxygen, so cells that don't get oxygen die, and so one of the specific ways that they die is in the absence of oxygen, the membrane that surrounds the lysosomes that keeps all that hydrogen in there ruptures. And so when those digestive enzymes then spill out inside of those cells, it causes the death of those cells. And so that is that self-eating that happens in the absence of oxygen. So autophagy is self-eating. So one of the things, like I said, that can trigger that is the absence of oxygen. Also certain toxins, so I thought this was cool. Cobra venom, the specific way that if you happen to get bit by a cobra, the specific way that kills you is that the toxins in cobra venom actually cause the breakdown of the lysosomes in your cardiac muscle. So the cells of your heart, the lysosomes inside of those cells will rupture, spill those acidic contents inside of the cell, and then cause the heart cells to die, which eventually stops the heart. So that's how cobras kill mice and the various other little things that they eat. Tay-Sachs disease, the book mentions it. I thought it was kind of interesting. So it's a genetic disease where the lysosome doesn't function correctly. And so basically, or more specifically, the lysosomes inside of the neurons, so inside of your brain cells, don't have a particular enzyme they need to break down a glycolipid, which is a part of the cell's membrane. And so basically then those glycolipids start to accumulate inside of the cell because you can't break them down, and they kind of clog up the cell, they cause it to swell, and they cause the neurons to die. So unfortunately, people who have that genetic condition, their nervous system doesn't function correctly, and typically that genetic condition is fatal before the age of 5. Vitamin A in excess. So vitamin A is one of the fat-soluble vitamins. So the fat-soluble vitamins being A, D, E, and K. But the fat-soluble vitamins can be stored. And so vitamin A, as opposed to like vitamin B, or any of the B vitamins or vitamin C, those are all water-soluble. Basically, if you take in too much, you get rid of them, right? And so that's why you see, like, if you're into energy drinks, sometimes I am, why they'll have this ridiculous amount of B vitamins in them is because the excess you just get rid of. But the fat-soluble ones, again, A, D, E, and K, it's easier to accumulate too much of them. And so if you have an excess amount of vitamin A, there's some evidence that interferes with the membranes of the lysosomes and then can cause them to rupture, which would then be consistent with the typical signs and symptoms that we see that correspond to vitamin A toxicity, which is that you get a lack of normal bone function, so bone pain, swelling, decreased density. So it interferes with the way that the osteoclasts function. And then you also see some nervous system dysfunction, so things like headaches and potentially some worse symptoms than that. All right, so that's it for organelles. And then in the third part of this, we're going to talk about producing protein. So we'll talk about DNA and RNA and protein production. So we'll see you next time.